So as you heard, I am a neurologist and neuroscientist at Penn. My chief area of research concerns how the brain is organized for visual processing. And I have spent, I think that's it, yep. So, and I am now entering my 20th year of working with brain imaging techniques as applied to the study of vision. As you heard from Professor Jones, I have the pleasure today of providing an overview of neuroscience and neuroimaging over the next 45 minutes to an hour. And one reason why this is such a pleasure is that I get to sort of summarize all these topics at really sort of a very broad and general kind of level, and then the team of outstanding speakers that come after me have to grapple them with all the messy details of actual science done actually in the lab and applied to real questions. An exercise like this of trying to summarize all of this material is really an exercise of forgivable lying. So I am going to have to, as I go through, simplify and omit and obscure lots of details, but hopefully for the goal of providing a basic heuristic understanding of a lot of these topics that I'm going to cover. In fact, actually, to make this a forgivable exercise for you and for me, as I go through, occasionally we'll take a step back from this high-level understanding that I'll be providing and pull back the curtain to show you all the messy, grubby details that are going on underneath the surface. But I promise it'll only last for a slide or two. I don't think there are any actual equations that appear with any of the slides, so this should be easygoing. But mostly I want to give you a sense of just at least the scope of the details that exist behind the generalities that I present. Okay, well, as you heard, and as Owen motivated very nicely in his remarks, neuroscience really has penetrated very deeply into many areas of social activity, far beyond the realms of science. In fact, actually, I think I found the ultimate demonstration of its reach. And so for many of you, if you were stuck in a ground stop on your flight on out here, you might have pulled the SkyMall catalog out of the seat back pocket in front of you. If you page through the SkyMall catalog, you'll find an ad for this device, which looks pretty handy. This is a large-scale magnifier, so there you are, retired, and you want to try and read a little bit of something. If you focus in on what this guy is reading with his large-scale magnifier, it is a functional MRI study with activity, it looks like in the medial portion of the cingulate gyrus. Now, why that would be something that your typical SkyMall catalog purchaser would want to make use of, I don't know, but I will say that for someone who works in neuroscience and neuroimaging, this is really the moment that I've worried we've jumped the shark. So, this might be the sign that my field is heading on back down when we're appearing in the SkyMall catalog. But, from this sort of ridiculous presentation to one which is more germane for this audience, and to add to the list that Owen provided, neuroscience evidence and these colorful pictures of the brain are appearing in courtrooms. And so here's another example. This is the case of Brian Dugan, who during the sentencing phase for his murder trial, the defense introduced neuroscience evidence provided by a professor and scientist, Dr. Kent Keel, actually friend of the MacArthur Law Neuroscience Program. And so what Kent had done was, Kent Keel performs neuroscience studies of psychopathy. So this behavioral state in which one is unable to sort of, at least summarized, make use of and feel the emotions of others, to not feel empathy towards others. And so Kent conducted both behavioral studies of the defendant Brian Dugan, as well as a brain imaging study. And at trial, Kent testified that the results of the brain imaging scan indicated that this man, Brian Dugan, is actually a psychopath. The brain imaging scan is consistent with that, and that therefore this should serve as some mitigating factor in his sentencing. Now, I think this is a matter for those here in the audience to decide much better than I how knowing that this man is a psychopath should or should not actually mitigate in a sentencing phase like this. But what I'm interested in for our purposes today is this claim, is the claim that you can take a brain imaging study that was obtained on this guy and then support the claim that because of some result that you find there, that this man is a psychopath. And we're going to be revisiting this example as we go through over the course of the next 45 minutes. And really build up bit by bit how you go from a scanner and someone sitting in there to the clanging of the machine to the processing of the data and ultimately a colorful picture like this, and then further on to the inference that this particular guy is a psychopath. Okay, so we're going to start really zoomed all the way out at the level of the organization of the brain. So I want to give you just a tour of the different lobes and structures and general roadmap of the organization of the brain. And then through this time, we're going to actually dip in and zoom in to look at individual neurons, the cells that organize, that are the basic organizational properties of the brain, and then examine their communication structure and how they work. So this is what we're going to do. We're going to go through a little bit both basic neuroanatomy, the connections and organizations of neurons and their function, and then see how that then applies to the use of brain imaging technologies and how it leverages these functions of the neurons to make measurements. Okay, so here's a view of the brain. This is the side view, so what you're viewing is the brain as seen from its left side. So the eyeballs would be up right around here. This is the back of the brain. And, whoops, wrong button. There we go. So what we can do first off is see that the surface of the brain conceals actually an awful lot of detail on the inside. So if we take a plane that passes through the brain along this axis, and then take a look at the structure of the brain viewed en face, you can see that there's a lot of further anatomical detail there, and we can provide some names for these things. So the first thing you notice about the brain is that it's wrinkly, right? It's all folded up, looks a little bit like a walnut. And so these wrinkles have names, so the surface or bump in the brain is called a gyrus, and the division or space in between each wrinkle is called a sulcus. The other thing that's quite obvious about the structure of the brain, if you view it in a slice like this, is that it has both a surface and a deeper structure. And so the surface is referred to as the cortex, which is this outer layer there. Cortex actually is from the Latin root for bark. So it's almost like the bark of a tree. It's the surface covering of the brain. So the cortical surface, which contains what's called the gray matter, and then the deeper structures of the brain, which is called the white matter. Okay? So there's the cortex indicated. It's the gray matter layer that's on the outside of the brain, and the white matter is deeper in. Okay, so from this most basic division of cortex and you have your gray matter and your white matter, you can now have an understanding of how nerve cells individually are organized in the brain and where they run. So let's take a look at that. So now we're zooming all the way in to look at a single nerve cell that makes up the structure of the brain. So one nerve cell, which is depicted here, has this very basic structure. It has what's referred to as a cell body, so that's kind of this bump down here, and then a long, long appendage that runs away from it. And so these things have names. The dendrites are these little spiny things that are at one end of the cell body. It has a very long process which extends from it called the axon, and that ends in a synapse. And so we're going to put these terms to work in just a few minutes to see how nerve cells connect together. So the first thing to know is that coming back now to this big picture view of the brain seen from in a slice preparation, is that nerve cells make up the structure of the brain, and in fact, the cell bodies reside up here in the gray matter, and the white matter are these long axon connections, okay? So here's a basic principle then of the organization of the brain. On the outside of the brain, in the gray matter, that's where the cell bodies themselves, the little chunks of these nerve cells are located. And what runs in the white matter are the very long connections that run from one nerve cell to the next, okay? And it's chains of these neurons together that make up the structure of the brain. Now, of course, already we've engaged in all sorts of unforgivable oversimplification, because in fact, the cortex is not just one thing. Actually, it's a many-layered structure, and there's lots of layers to the cortex with lots of different neurons at different layers, and they're all interconnected. And in fact, there's not just one sort of cartoonish sort of neuron that I showed, but there's actually dozens and dozens and dozens of different types of neurons, and each neuron has a slightly different sort of structure with different kinds of dendrites and axons and ways that it's connected together. But we're going to ignore all that. Let's go back to our cartoon. So here we are back to our nice, comforting nerve cell. So here we again have a neuron. Here's the cell body, right, with the dendrites up here and an axon. So the dendrites and the axon, this is the basis of the communication between one nerve cell and the next. And this is really then the basis of what it means to have an active nervous system and to engage in thinking and operation and moving and emotional states. It's really all these are all manifestations of the electrical activity of nerve cells and their connection together. So let me give you a sense of how that works. Okay, so let me introduce a new term. This is called the action potential. The action potential is an electrical discharge that travels down the axon. That's the communication going from one nerve cell to the next is something called the action potential, an electrical discharge. So how that works is the following. So each communication event of a nerve cell begins with a nerve cell receiving input at its dendrites. So this is where information comes into a nerve cell at the dendrites. So the dendrites receive excitatory information from other nerve cells. That excitatory information triggers off an action potential, which is an electrical wave of activity that travels down the axon, right, towards this other end, and down here what are called the synapses, okay? And so then what it means to then have a brain and sort of all this activity is to have a chain of neurons all hooked together where the axon of one neuron connects to the dendrites of another neuron. And this is the basis of this communication. You have lots of neurons hooked together, one axon connects up to the dendrites of others, and of course this becomes incredibly complicated because you can have multiple axons, multiple neurons converging on one neuron, and that neuron can then connect off to another one, so you have multiple levels of convergence and divergence of neurons connecting together. Where the action really happens, it's going to be relevant for a lot of things we talk about, is here at the synapse. So the synapse is the point of connection between the axon of one neuron and the dendrites of another. All right, so we're going to zoom on in now and see what's happening right at this point of connection between two nerve cells. Okay, so here we have a real blown up view. So here comes the axon of neuron number 1, and that ends in a little bump. And then here's the dendrite of neuron number 2, okay? And one interesting thing about these connections is that there's a little gap. They never actually quite contact one another. They get very close, but the axon of one nerve cell doesn't actually make physical contact with the dendrite of number 2. There's a small gap there. And now what I'm going to tell you about is how information crosses that gap, okay? Down at the end of the axon, there are little bubbles called vesicles, and those vesicles contain neurotransmitter. So neurotransmitter is a chemical, which the body makes, that it uses to communicate from one nerve cell to the next nerve cell. Okay, so the vesicles, these little bubbles, they have the neurotransmitter, these are chemicals, and on the dendrite, right, so this is neuron number 2, the dendrite contains little receptors. And so you can think of these things as baseballs and catcher mitts. There's a little bag of balls here and a whole bunch of gloves waiting to catch them. And so what's going to happen is that the neuron number 1 will communicate to neuron number 2 by throwing a whole bunch of balls which get caught by the receptors. And so the way this works is that an action potential, right, that electrical discharge that has moved down the axon, arrives at the very end of the axon and it kicks those balls down to the very edge where they release the neurotransmitter, which then diffuses across the little gap and gets caught, right, by those receptors. And then when they get caught by that dendrite, this then triggers an excitatory event. So this is now an excitatory electrical event in this dendrite, which then triggers its own action potential, which zooms off to the next one. Okay? So the important message here is that neural communication has two phases. There is a rapid electrical phase that zooms down the axon. That's the action potential. So it goes very quickly down this very long cable that connects one neuron to the next. And when that electrical event zip, reaches the very end, it triggers this release of the neurotransmitter, which slowly diffuses across the tiny gap, gets caught by the next neuron. Oh, time for another action potential. Zoop down the next axon. Okay? So rapid electrical, slow chemical communication, then rapid electrical. Right? So two components. Again, I have engaged in an enormous oversimplification. So it turns out that these neurotransmitters, right, this key chemical element that carries information from one nerve cell to the next nerve cell, well, these neurotransmitters come in many different varieties. In fact, here's some examples of these. And some of these terms might even be familiar to you. So you might recall serotonin as a neurotransmitter or chemical you've heard of before, which has been blamed as the reason why after eating lots of turkey after Thanksgiving you feel sleepy. Serotonin is also involved in a lot of the antidepressant medications. You've heard of selective serotonin reuptake inhibitors. These are a class of antidepressant medication that have their effect. It's kind of complicated, but to a first approximation upon levels of serotonin in the brain. You've heard of dopamine, and you'll hear more about dopamine today. This is a neurotransmitter which is engaged in lots and lots of different things, including movement and coordination of different functions in the brainstem. But is also involved in reward systems, generally speaking. You're going to hear a lot about reward systems when you hear about addiction. Norepinephrine, this is another neurotransmitter, which, part of the reason why cocaine makes people shaky and excited is because it's increasing the potency of norepinephrine within the nervous system function. So there are all of these different neurotransmitters. They all have slightly different distributions within the brain. They're produced in different spots. They have different kinds of effects. And furthermore, on the receptor side, on the catcher's mitt side, the story becomes complicated. Some receptors on the dendrite are excitatory, but some are negative or inhibitory. And yet others are both excitatory and inhibitory and have two different kinds of neurotransmitters connecting at different points. So again, enormous complexity that lies beneath this simple model of rapid electrical communication followed by a slow diffusion of a chemical. And a lot of the important details about those neurotransmitters end up having very important clinical implications. And we're going to be returning to those over the course of the lectures you'll hear over today and tomorrow. Okay, so we have that idea. Neurons communicate to one another, electrical activity down the axons, chemical communication here at the synapse, which then triggers off the next action potential, and you have these networks of neurons that have propagating waves of neural activity. So here's the thing, all of this sort of generating electrical impulses and releasing neurotransmitter and then sucking it back up and then releasing it again, all of this requires cellular machinery to be operating, and that cellular machinery requires energy. And so the source of this energy is through metabolic activity, right, through the functioning of the nerve cells that takes place here in the cell body. So the cell body, the reason this is a big bump here, the reason the cell body has sort of one big lump and then the long axon is that cell body is where the factory is that's responsible for processing energy and making the proteins that make the cell work and recharging its electrical potential and all the rest. And so to do that, the cell body requires energy in the form of glucose. That's the formula for a glucose molecule, right? So glucose is a simple sugar, right, which appears in lots of things we eat and also is the product of the breakdown of the things that we eat inside the body to produce glucose. And so that glucose is combined with oxygen, and that gets effectively burned within the metabolic furnace of the inside of the cell, and that's what's providing the energy, okay? So, in addition to having rapid electrical communication down nerve cells, and then a slow chemical communication from one nerve cell to the next, neurons are also metabolically active. They're burning glucose all the time to support these operations. And in fact, the brain, even at so-called rest, when you're just sort of daydreaming or relaxing, is enormously energetically active. It's consuming about 20%, right, of the blood flow that comes up through the entire body is going up to the brain. An enormous amount of the caloric requirements that we have daily is just to support the constant buzzing activity of nerve cells. So it's an enormously energetically costly activity to keep this network of communicating nerve cells running. Okay, so if you would like to take away just a couple of key points so far, here's what we have. So we have the brain divided into the cortex, right, which is the gray matter, and the white matter. The cortex or the gray matter contains the neuron cell bodies. The white matter contains axons, which are the connections between one nerve cell and the next. The cell bodies consume glucose and oxygen, right, and neurons communicate electrically and chemically. And there are tons of fascinating and important details about all these things. Just one little tidbit, right? So the gray matter, or the white matter rather, which has those long little connections between one nerve cell and the next, well, boy, that is a key structure which is damaged in traumatic brain injury, right? So if you have someone who has a concussion and a closed head injury, the brain is sort of a big soft jello-like substance that then twists, and those tiny microscopic little axons that connect from one nerve cell to the next, those can get sheared and broken, right? So just for example, part of what makes a traumatic brain injury cause damage is the breaking of those tiny little axons that connect one nerve cell to the next within the white matter of the brain. Just one little tidbit. Okay. So, now that we have a sense of these different sorts of connections and cells that make up the structure of the brain, I'd like to give you an overall picture of the different territories and regions and then their functional assignments, okay? And again, this is going to be an exercise in oversimplification, but I think this is a fairly useful roadmap for understanding how different sorts of mental and emotional states are divided in function across the areas of the brain. Okay, so first let's talk about lobes and hemispheres. So again, here's our side view of the brain. So back of the brain here, your eyeballs would be up right around here, your ears right around here. So that's the side view. We can take the brain and sort of flip it over, whoop, and see the medial view. So normally you can't see this surface of the brain because you have two hemispheres and they're stuck together. But if you were to cut the brain in half and take the two hemispheres apart and then look at this half that you've just separated away, what you'd see is this surface, the so-called medial or mesial view of the brain, okay? So By tradition, the different areas of the brain are broken up into so-called lobes, right? All of you have heard of frontal lobes, for example. And so these different lobes have, generally speaking, different sorts of functional properties. So let me give you a sense of that. So the occipital lobe, which is in the back of the brain here, remember eyeballs up front here, this is the very back of the brain. Eyeballs would be up here, this is the very back on the other side view. This is an area that's involved in vision. So this is where most of the nerve cells that are concerned with processing visual information are located. There's another lobe, the parietal lobe, that sits up here. This is an area of the brain that's involved, generally speaking, with attention and spatial representation, knowing where things are in the world. Now and again, there's lots of other stuff that these parts of the brain do. In fact, there's a little bit of language stuff that goes on in the parietal lobe too on the left side, but we'll get back to that later. But generally speaking, the parietal lobe up here, very concerned with maintaining a map or a representation of where things are in the environment. Okay, the frontal lobe, this humongous piece of brain, which is a distinctive feature of the human brain. So our primate relatives also have frontal lobes. In fact, mammals generally have frontal lobes, but boy, we've got an extra big frontal lobe. This is a very large area. There are, I'd say that this is a region of the brain in which there is great work going on to try and elucidate all of the functions that it serves. So this, again, is a gross oversimplification, but generally speaking, the frontal lobe is involved in language production, right? So production of symbolic thought and language, and planning, right? And inhibition of prepotent or automatic responses. You'll be hearing more about that. And then finally, the temporal lobe. There's lots of stuff that goes on here, but the piece that's going to be important for lectures that you hear later on is that this portion of the temporal lobe here is very critical for memory. Damage here results in someone who is unable to learn or remember things. Or, I'm sorry, is unable to learn information that they can maintain. Okay. So beyond this sort of division into different chunks, it turns out that you can divide the brain pretty effectively into a left, right, front, back, top, bottom division, and have a pretty good idea of what's going on in these different areas. So all of you have heard about like left brain, right brain. Let me see if I can disabuse you of some of what you might have heard about left brain, right brain. Okay. So we begin with this. It's very obvious we have a left side and a right side of the brain. Maybe there's some different functions that are attributed to these different parts of the brain. And as it turns out, there is. So generally speaking, for almost everybody, there are a few left-handed people out there for whom this is swapped, but not all the lefties. The left hemisphere of the brain is involved in language and the skilled use of the hands, like how you know how to use a tool, for example. Okay? Generally speaking, that is the left hemisphere function, whereas the right hemisphere is more specialized for spatial representation and attention. That's it, right? Actually, not this whole creativity thing or drawing upon your left side of the brain and stuff. We'll come back to that. There's another division between the front and the back of the brain. There's a big sulcus here, one of these grooves in between the folds of the brain, the central sulcus, and that divides effectively the brain into the front and the back halves. And generally speaking, the back half of the brain is concerned with perception, that is bringing information in from the sensory world and processing it. And generally speaking, the front half of the brain is concerned with action, producing movement, producing language, producing output. Okay? And interestingly enough, this is a division that runs all the way down the neuroaxis, even down into our spinal cord. And then a final division is between, in the back of the brain, between what and where. It turns out the top half of the brain here is concerned about where things are in the world, and the bottom half of the brain back here is concerned about precisely what things are. Who is that guy? What color it is that I'm seeing? Which one is my cup of coffee? That's all going to be down in ventral as opposed to where, which is where things are positioned in the environment. There's a final division that I want to mention, and that's broadly speaking between what's referred to as the cortical, or the upper portions of the brain, and subcortical structures. These are structures that are deeper inside the brain. Broadly speaking, some of the subcortical structures are more evolutionarily ancient, and these structures are engaged much more in sort of automatic or reflexive behaviors and properties of our nervous system, things that you do reflexively or quote-unquote unconsciously, whereas the cortical structures are more engaged with deliberate action and planning, generally speaking, and you'll hear more about that. Okay? So to summarize, there's regional specialization for mental function within the brain, okay? And see if we can discard a couple of these ideas. We only use 10% of our brain. I'm so creative, I'm right-brained. No, please don't say any of those things. There's absolutely no basis to this whatsoever. We're actually using all of our brain all the time. There is some regional specialization where certain sorts of mental activities seem to require the function of parts of the brain a little bit more, but it's not the case that you have hidden latent powers if only you could tap into the other 90%. Sorry, I'm not even quite sure how that got started. Okay. So now that we've done our tour, we understand the brain on a microscopic level, right? Its communication, electrical and chemical, and its metabolic function, and we understand its broad regional organization. Now let's take the brain and we want to put it under a microscope. Let's figure out how we're going to actually make measurements of the function of the brain, which are relevant to some of the questions that are going to be discussed over the next couple of days. So the first division I'd like to introduce you to is making measurements of the function of the brain using its electrical properties or its metabolic properties, okay? And this comes back to then these basic functions of nerve cells that we talked about before. So there's one class of imaging approach, one class of ways to make measurements from the brain functioning, which is based upon its electrical activity. Okay? So all of these neurons engaging in their action potentials and releasing their vesicles and sucking them back up, all of that activity generates an electric field, and that electric field can actually be measured, right? It can be measured on the surface of the skull and on the surface of the scalp. And so these techniques have been around for a long time. EEG, or electroencephalography, is a technique of placing wires directly against the skin and making measurements of the electrical activity of the brain. And this has been around since the 1920s and allows you to make measurements which are outstandingly resolved in time, because these electrical fluctuations you can measure microsecond to microsecond, but unfortunately are kind of fuzzy in space, because by the time those electrical signals have made it through the insulating properties of the skull, they're very smeared on out. So a typical image or measurement that's made using these electrical techniques has very good resolution in time, but is kind of a smeary picture in space about where it's happening in the brain, okay? EEG is the technique that's been around for a long time. There's a modern, much more expensive version of this called magnetoencephalography. This is a really cool kind of space-age technique involving superconducting quantum interference and liquid helium-cooled detectors and this big device and multi-millions of dollars, and it's a little bit better than this. So, but you know, for millions of bucks, you can get a slightly better picture you're going to do. So, okay, that's electrical imaging. There's another sort of imaging approach, which is metabolic imaging. Okay? Metabolic imaging is measuring properties of the nerve cells related to their metabolic factory activity, right? Consuming of glucose, consuming of oxygen, needing more blood to support these operations. Okay? So there are some techniques which are nuclear medicine techniques, PET and SPECT, so positron emission tomography, single photon emission computed tomography. And these techniques involve providing a radioactive version of a metabolic substance that the brain uses, right? So you take sugar and you replace one of the carbon atoms in the sugar with radioactive carbon, and you inject that into somebody's blood, and that radioactive sugar makes its way up to the brain, and the brain cells, hungry little fellows that they are, take up this glucose and they start chewing on it to support their metabolic activities. And when that radioactive carbon undergoes radioactive decay, it releases energetic particles that go flying through the brain tissue and out the skull and are caught by detectors which are arrayed around the brain. Okay? And there's a few of these techniques which have as their basic idea that you're going to create a radioactive substance which makes its way up into the brain, and then you measure energetic particles released by the decay, the radioactive decay of that substance at different locations. And of course, wherever more glucose is being taken up, right, that's where the neurons are active and hungrier, and so you'll get a larger radioactive signal coming from that area, right? You can imagine the most simple version of this experiment would be you inject the radioactive sugar, and then you put a Geiger counter over someone's skull, and what you'd measure over time is right, as the glucose comes up, it engages in this radioactive decay, and then goes away. And in fact, instead of just having one Geiger counter, you have an array of Geiger counters around the brain to make these measurements. Okay? That's PET and SPECT. There is another technique which we're going to come to in greater detail in a moment, which is functional MRI, which is making a measurement about blood flow. We'll talk about that more in a second. All of these techniques have the property that they have poor resolution in time, because this metabolic activity and the flowing of blood, this is a relatively slow thing, but good resolution in space. You tend to get pictures with better spatial resolution. Okay? Just again, pulling back the curtain for some complexity, just so you know about it, PET imaging is actually a really critical technique for measuring certain metabolic properties of the brain that we're not going to touch upon anymore in this lecture, but might come up later. It turns out you can use PET imaging depending upon what you stick your radioactive compound onto. You can measure lots of different things. For example, you could take a neurotransmitter like dopamine, right? So that chemical communication between one part of the brain and the next, and you make some radioactive dopamine, technically L-dopa, and you inject that in, and then you can measure where there are cells in the brain that are avidly taking up dopamine, for example. And these sorts of techniques are important, actually, in a lot of studies of drug addiction. You'll hear more about that. And so you can use this to measure, basically, the distribution of all sorts of stuff in the brain. Okay, we now come, finally, to bold fMRI. And so this is really the key thing that we're going to discuss for the next few minutes. So of all the techniques I've talked about so far, we're now getting to the number one grand poobah of all brain imaging techniques, bold functional MRI. It's been around for about 20 years. This is a technique of measuring, again, non-invasively, the neural activity, the relative activity of nerve cells in the brain using an MRI scanner. I'd like to walk you through how that works. And basically our first task here is to understand how we go from activity of nerve cells, right, their electrical and chemical activity, into creating an image of the brain that reflects that electrical and chemical activity of the brain. Okay. So we're probably all familiar with MRI scanners. In fact, I bet that many folks in this room have been inside an MRI scanner just for routine diagnostic purposes. And in fact, there are some of you in here who later this afternoon or tomorrow, I understand, will be getting in the scanner for the purposes of engaging in an experiment. So an MRI scanner is able to produce anatomical or structural pictures of the brain. And in this function has revolutionized a lot of the practice of neurology and neurosurgery for its ability to provide incredible detail and clarity of the different anatomical portions of the brain. In fact, there was a Nobel Prize given out for this work. Depending upon the way that an MRI scanner is used, it's able to create lots of different kinds of pictures of the brain, right? It can create images which are able to show you where the blood vessels run in different parts of the brain. It's able to create an image which measures where there's blood that is leaked out of blood vessels, and these are all things which are related to its clinical use, its use in medicine. It's able to detect where strokes have happened, using what's called a diffusion-weighted image, and even able to detect sort of the white matter spots of damage that might have been the result of multiple sclerosis or bad traumatic brain injury or things like that. So lots of different ways of doing it. But these are all measurements that are made of the structure of the brain, and what we're interested in here is function, right? The electrical and chemical activity of nerve cells moment to moment. And the key for understanding how this is possible using an MRI scanner is to know about two fortuitous properties of the brain and physics. So the first fortuitous property is something called neurovascular coupling. And so here's what neurovascular coupling is. The brain is a whole bunch of nerve cells, right? This big bag of nerve cells with tiny blood vessels that are feeding blood to all these different portions of the brain. And as I told you before, the brain is very hungry for metabolic substrates. It needs a lot of glucose, needs a lot of oxygen, and needs a lot of blood coming through to flush out the waste products of that metabolic activity. So there's an incredibly detailed and ramified network of blood vessels that then feed to every corner of the brain. And there is an intimate relationship between those nerve cells and the tiny blood vessels that feed that piece of the brain, such that when the nerve cells increase their activity, when they become more active, when they take up more glucose, when they release more neurotransmitter, they send a signal, which by the way is still the subject of active investigation, what this signal is, but they send a signal to the local blood vessels that causes the blood vessels to dilate, to get bigger, and bring in more blood. Okay, so that's the basis of neurovascular coupling. Where the flowers in the brain are thirstier, the garden hose is made wider to provide more water. That's it. And what's really great is that these things are coupled on quite a small scale, right? So a point of neural activity gives rise to an increase in blood flow, which is tiny, tiny, smaller than the tip of my pinky, and I'll show you that in more detail in a moment. That's fortuitous property number one. Here's fortuitous property number two. As you know, our blood is made up of red blood cells, and so red blood cells contain a protein called hemoglobin. And hemoglobin is the key oxygen-carrying molecule in our blood. This is the thing in our blood which grabs oxygen from the lungs and delivers it to the tissues in the body where it's needed. And the way that that works is that hemoglobin has at the very center of this big glob of protein an iron atom. And effectively, the way that hemoglobin carries oxygen is that it brings this iron atom into the lung and allows it to rust. The iron atom grabs oxygen. It becomes a rusty iron atom. That's what rust is. It's iron that has been oxidized with oxygen. And then that rusty iron atom is carried into the body, and then it releases the oxygen and goes back to a pure state of iron. And that's it. That's what hemoglobin is. It's carrying around these iron molecules. And what you know about iron is that it is a ferromagnetic material. In other words, if you have a piece of iron, you can stick a magnet to it. Okay? And finally, it turns out that rusty iron is slightly less magnetically attractive as compared to unrusty iron. It would be hard for you to measure this effect using just a magnet and trying it out in your kitchen, but it nonetheless is a measurable property that rusty iron and not rusty iron is slightly different in its magnetic properties. So here's how we then put it all together. What's an MRI scanner? An MRI scanner is a massive magnet, okay? So there is a coil, a loop of wire, which surrounds the person that's been slid into that scanner. And this is superconducting wire, which is cooled by liquid helium, which then has current running through it. And what you might remember from high school physics is if you have a coil of wire and you run an electric current through it, you create a magnetic field. So there's this massive magnetic field. And so within that magnetic field, you can measure the local perturbation, basically the wrinkling of a smooth magnetic field that your machine has made by the presence of these iron atoms. Okay? And the slight difference between iron atoms that are carrying oxygen or not carrying oxygen is measurable using this system. Okay? So to summarize an awful lot of stuff, if you have increases in neural activity, that leads to changes in local blood flow. So just local in the brain where the neural activity has taken place, there's more blood flow. This changes the relative concentration of rusty iron, which changes the local magnetic field, which is something you can measure with your MRI scanner. Okay? There's this chain of physiology and physics that links together changes in neural activity with changes in a picture of the brain that's made of it. Okay? So now let's take a look at what the structure of these pictures are. So here is a typical raw image slice. It's actually a couple years out of date, but a raw image slice of what the data from a functional MRI study of somebody might look like. So this is one slice through the brain, a horizontal or axial slice through the brain at one point in time. And this image is made up of lots of tiny, tiny little points, right? So if you got in real close to a TV, it's made up of tiny dots. Well, in the same way, a picture of the brain is made up of tiny little measurements, which are referred to as voxels. So a voxel is the smallest unit of measurement that makes up these pictures. The reason it's called a voxel is because it has volume. It's a 3D cube that then, when assembled, makes up the entire volume of the brain. In typical applications, these voxels are somewhere between 1 millimeter and 3 millimeters on a side. In the 3 millimeter size, that contains about 600,000 neurons. Okay, so one of these little boxes, these are cartoonishly big, but one of these tiny little boxes that make up an image of the brain contains on the order of 600,000 neurons. If you make your voxel smaller, you can get that down to about 25,000 neurons. Just to give you a sense of that size, here's a finger, and here's a little 3 millimeter square on the tip of it. Okay, so that's the size with which a typical neuroimaging study is measuring the activity of the brain at point by point by point. And then these images are acquired, one brain image every 1 to 3 seconds over time. Okay, so you get brain picture, brain picture, brain picture, brain picture over time. And that then is the raw data that makes up a functional MRI study. Okay? And so then the final thing I'd like to talk about is how we go, from this raw data over time to a processed picture of the brain in action. All right, and we're going to do it by returning to this guy and this particular claim that functional MRI shows that Brian Dugan is a psychopath. So here we go, the claim. Let's start with this. Psychopaths show reduced responses to emotional stimuli within the amygdala. The amygdala is a deep brain structure which is involved in the processing of emotion, amongst other things. All right, so what does that look like? So you have your brain, you have your one slice of the brain like this. We're actually going to acquire multiple slices up and down through the brain to get a whole three-dimensional volume. And so you take your person and you put them in the scanner, and over time, while the brain is taking pictures, or the scanner is taking pictures, beep, 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 so making these measurements, the subject is looking at a screen, and on that screen they're shown pictures of faces. They're shown a fearful face, and then a face which allegedly does not have any emotional content. He looks kind of angry to me, actually. And then a fearful face, and a neutral face, and a fearful face, and a neutral face. And so your subject, you take Brian Dugan, you stick him in the scanner, and you show him these faces, emotional faces and faces without emotion. And what you do is you measure from one point in the brain the signal, right, that fMRI signal that rises and falls over time. And what you might notice in this cartoon example is that when the fearful face comes on, the signal rises, and when the neutral face comes on, the signal drops and rises and falls and rises and falls. Okay? That's what the raw data looks like. And then for each point in the brain, you can quantify how much of a difference was there in this brain signal measurement between the fearful faces and the neutral faces. How much more active were the neurons when they were looking at faces which showed fear versus neutral faces? How big or reliable was that difference? And you can quantify this many different ways. In typical practice, it is expressed as a statistical effect, the size of a statistical effect. Now, that's the measurement of how much fearful faces changed brain cell activity at that one point in the brain. So we could write down, say, that that was a 2.5 size effect at that location. And you could do that calculation for each of these voxels, for each point in the brain. And you could just write down the number in these little boxes, but that would be very tough to read. So a typical shorthand that's used for these is instead of actually writing down the effect size at each of these points, you color code it. So you make a small effect be red, and you make a big effect be yellow, and you color code each point in the brain. So that's just shown for one voxel, for one point in the brain, but you have these measurements from over the entire brain. So ready? Poof, we'll do this for every point in the brain. Every point in the brain gets a color. And that color indicates how much of a neural activity change was associated with seeing a fearful face versus a neutral face. All right? So then that's now a processed image. But then we still have some ways to go from that processed image to the final thing that might turn up in your courtroom. And this is where we enter the realm of what I'm going to tongue-in-cheek refer to as neuroaesthetics. Okay, here's our image after it's been processed and we've assigned a statistical value to every point in the brain. Well, the problem here is that every point in the brain is assigned a statistical value. And we know that some of these are, not actually significant, that just by chance, the random just fluctuation of blood flowing back and forth, that there's going to be some kind of value here that wasn't really related to seeing the fearful faces. And there's perfectly good statistical techniques to be able to tell how big an effect there should have been to be able to tell the difference between a real effect and just some noise that turned up. And so you can calculate what's referred to as a threshold. You say, okay, only an effect bigger than X, than this size, we'll call real, and everything else we'll say was just noise. So let's apply that threshold. Okay, better. Now we're just seeing those points where there was a big enough effect that I can trust it, that I believe that it's true. Well, part of the problem, though, is that all of these little dots that make up this image are scattered all about. And it turns out that for perfectly good statistical reasons, it's actually beneficial to smooth these images in space a little bit. If you're looking for an effect which is spatially extended, you do better to detect those things. This is just basic statistical approaches to these data. If you smooth the data in space, to engage in digital smoothing. So that looks like that. We take these data, smooth it in space, and now we have one blob. It's easier to detect the big blob, but as a consequence, the small dots, they're harder to find. But that's a sacrifice you might accept if you want to be better at detecting the big stuff. All right, well now we want to try and figure out where in the brain is that blob. Well, that's kind of tough. This brain image that I've got it displayed on is kind of fuzzy. That's the raw fMRI image. But you know what? That scanner that I used to get that functional data, I can use that to get a nice high-resolution anatomical picture of the brain too. Maybe what I can do is display this activity, right, that statistical measurement, I'll display it on that nice anatomical image I have. That's better. Now I can see precisely where this blob of activity is in relationship to the structure of the brain. But I still have a final problem, which is that the brain is a volume, right? It's got an extent in space, and on a piece of paper I can only show one slice through the brain. That's kind of a limitation. So maybe what I can do is go talk to my friends over in the graphic arts department and do a three-dimensional reconstruction of the brain to show where this blob is in relation to the entire three-dimensional structure. There we go. So now what I finally have here is a three-dimensional view of the anatomy of the brain with a cutout so I can indicate where the blob of activity is. Okay? Now let me emphasize, every step along the way here is a perfectly rational and very well justified process that you might apply to the data. None of this is chicanery, no one's trying to fool anybody. These are well-grounded statistical and display approaches. But nonetheless, look at the distance that we've traveled from the original sort of image in its coarseness to then the final polished result. Just to give you a sense then of how these things come about and how they're made. There's another problem we need to deal with. These are the data from one person. Suppose I want to make a claim about what's true about psychopaths in general, okay? I might collect these data from 20 people who are psychopaths. And then I have the problem of kind of getting all their data together. Well, then we have to grapple with the problem that every person has a slightly different shaped brain. So here might be the results from doing this analysis, from taking 10 different people and putting them in the scanner and showing them pictures of fearful faces and neutral faces. And, it looks like it's pretty consistent. Most folks have a blob that's up here in this region. Some of them it's not really there, and some of it's moved around, but generally speaking, there's something up there. Well, but I need a way of trying to combine this information across subjects, and there's a very well-developed set of techniques that allow us to register the brain of one subject to another, basically to treat the brain like a piece of silly putty so that we can bend it and mold it a little bit to match them up across the group. All right? So you can create an average representation of the brain and then show where that activity is located in average across different people. Okay? And again, there are some enormously sophisticated techniques for doing this kind of thing, and I'm just showing you a cartoon example. But generally speaking, what's done is you need to get those 10 subjects, their brains, into some registered group space and then combine their data to find what's true on average for the group. So we're now finally in a position to return to this core claim that psychopaths show reduced responses to emotional stimuli within the amygdala. So what actually then was done was that you put people in the scanner one by one and showed them fearful faces versus neutral faces in this example, and measured the difference in neural activity at each point, and then combined the data across people. And so what you might find is that here in the amygdala, if you did this for 10 psychopaths and 10 normal people, that you'd find that there was actually a greater amygdala response to fearful faces than seen in the psychopaths. And this might comport fairly well with what you believe is true about psychopaths, that they have a blunted emotional response to other people's, say, fear and emotional states. So the amygdala, a region of the brain that's involved in emotion processing, has a greater response to seeing other people looking afraid for normal people than it does for psychopaths, okay? And this claim might be quite well supported, that if you do the statistics, there is a significant difference between these groups. But what these bars illustrate here is the range that was found in each population. And again, this is a cartoon example. But what's shown is that while there is a difference on average between psychopaths and control subjects, nonetheless, there is overlap between the two groups. And that overlap becomes really critical for asking this claim, or making this claim, that this person is a psychopath, because he has reduced responses to emotional stimuli. So his response might be right here, right? Perfectly typical of the psychopaths, but also overlapping with that of normal people, okay? And let me hasten to add that this is not a revelatory insight about that only neuroimaging has. This is something that I'm sure all of you have dealt with the provision of scientific evidence in your courtrooms for years, that claims are made in general about populations that you then must translate to specific people. And this is, I think, laudably why this is an area of focus that Owen mentioned before for the Law and Neuroscience Project. But I just want to illustrate, just make quite concrete, that just because it's a brain imaging result, and just because it's a brain imaging picture, it is not excluded from this core inferential limitation that any kind of claim like this would have to be subject to. Further, there's a deeper issue. And the deeper issue is, if psychopathy is defined by behavior, how is the neuroimaging data even relevant? Okay? So if what it is to be a psychopath is to have a certain pattern of behavior responses, right? That if I can measure psychopathy and say, oh, you're a psychopath, but you're not, based upon behaviors that you make, how does it even help you or tell you anything to take a look at the brain structural or brain functional responses in psychopaths versus normal people? Basically, maybe everything that you need to know about assigning people into these categories can come from the behavior, okay? And that final area, how we take these brain images and then apply them to inferential questions, is something that I'd be happy to talk about a little bit more during the question and answer session that would come up. But I'm going to stop there, and we'll bring Owen on back and have a discussion about these topics. Thanks very much for your attention.